This is podcast 29. All right, we're going to move into mitosis and meiosis. This is eukaryotic cells, and we're going to talk mostly about animal cells and eukaryotic cells. So mitosis is part of the cell cycle. If we consider the cell cycle as the entire lifetime of the cell, a map of the entire lifetime of the cell, mitosis is just a small slice of that lifetime. Some people get confused and they think mitosis is the cell cycle, but cell cycle also includes the first growth phase, the S phase, which includes DNA synthesis, and the second growth phase. Altogether, these are called interphase, and then mitosis is separate from that. Cells might stay in G1 for most of their life. These are differentiated cells in an adult, for example, and they would stay in the first growth phase and for most of their life. If so, that's called G0. And then cells that don't do that, but instead go through growth phase and then decide to duplicate. First, they have to duplicate their DNA prior to mitosis. Once they are committed to the S phase, they will continue through G2. That's why the second growth phase is usually shorter. And then on to mitosis. Some cells will hang out in G2. We'll talk about some special cases. But most of the time, once you're committed to go to S, you're going to go all the way through mitosis. In eukaryotes, of course, they have linear chromosomes, and it's important to know the difference between chromosome and a chromatid. So a chromosome in a eukaryote or a prokaryote is anything that you could, if you pulled on it, you get a whole structure, one whole structure. So if I pulled on this sort of thing, I would get this whole structure. And if I pulled on this sort of thing, I would get this whole structure, one whole structure. They aren't exactly the same, but you can see it's all connected. So before S phase in eukaryotes, chromosomes look like this. They have a constricted area in the middle. This is the centromere. It's not always in the middle. It can be up higher or lower, and that's how we can identify individual chromosomes. But there is one arm of DNA associated with every centromere. After the S phase, an exact duplicate of that DNA is made. And now this chromosome, single chromosome, has two chromatids, one here and one here. Let's see if I can use my fancy new toy here. So this thing here, that's one chromatid, and this thing here, that's another chromatid, and they're all together one chromosome. Each of those chromatids has exactly the same sequence as the other one. So it's an exact copy. This is different from homologous chromosomes, where you have one from mom and one from dad. Those aren't exact copies of each other, but they are similar to each other. So at a given position on a chromosome, let's say right here, that is the location of an eye color gene. In homologous or in sister chromatids, that same location on the sister chromatid would have exactly the same sequence. If it's ACCG on this chromosome, it's also ACCG on that chromosome. If we were looking at two homologous chromosomes, two versions of chromosome number 2 in humans, one of those would have a sequence that codes for, say, blue eyes, and one may have another sequence that codes for brown eyes, in which those two gene sequences may be slightly different. So maybe it's ACCG on one locus of one chromosome and ACCA on the other locus on the other chromosome. Okay, so once again, before S phase, this is you have a single chromatid per chromosome. After S phase, you have 2 chromatids per chromosomes, and humans have 23 pairs of chromosome. And don't get confused about that word pairs. You want to think about pairs like pairs of socks. So if you have a pair of socks, you have 2 socks. So humans have 46 chromosomes, 23 pairs. 2 versions of chromosome 1, 2 versions of chromosome 2, 2 versions of chromosome 3, etc. You get 1 from mom, 1 from dad. But 46 total. Okay? Before the S phase, each one of those chromosomes looks like this, and after the S phase, each one of those chromosomes looks like this. Okay, just walking through what animal cells look like, and then a picture diagram before, just following a couple chromosomes, not the whole human complement of 46 chromosomes, and what is happening in these different parts of mitosis. So during interphase, over here, again, interphase is the first growth phase, G0 or G1. Synthesis, S, and the second growth phase, G2. All of that is considered interphase. If you look at a cell in that situation, it would look like this, in that you see an intact nuclear envelope. You see diffuse chromatin. You can probably usually see a nucleolus. And then you see 2 centrosomes with centrioles in them if this is an animal cell, and these are animal cells in this example. Above here, blue is going to be DNA, so chromosomes are going to be blue. Green is stained, those are microtubules, and red is actin. Okay? During prophase, what's going to happen is the nuclear envelope is going to start to degrade. The centrioles are going to start to migrate to opposite poles. Okay, you see that here. And you'll start to see DNA condensing into chromosomes. So this is very diffuse in the case of interphase, but now you can start to see individual squiggles, which will become chromosomes, and that's going to happen in prophase. So blue is the DNA, that's going to be chromosomes. Green is microtubules. In pro-metaphase, just before metaphase, you see that individual microtubules are lining up and attaching to individual chromosomes at their centromere at a protein complex called the kinetochore. We'll talk about that in detail. And then they will start to move back and forth in the cell until they reach metaphase, where they are lined up in the middle. So this is always what it looks like in a diagram, where they're all lined up in the middle, nice and neat like that. In a real cell, these chromosomes are longer than the axis that they're lining up on, and so you get squiggles that sort of bleb out from the middle line, but you can see that there's definitely a middle line here. And so these chromosomes are lined up in the middle, middle. That's how you can remember that. And then during anaphase, what was a chromosome with 2 sister chromatids? Okay, here's 1 chromosome, 2 sister chromatids, 1 chromosome, 2 sister chromatids, and so on. During anaphase, what happens is 1 chromosome splits in half and becomes 2 chromosomes. So now this is a chromosome and that was a chromosome. And during metaphase, this was all 1 chromosome, and 1 becomes 2. Splitting down the centromere, 1 chromosome becomes 2 during anaphase. And then the separate chromosomes will start migrating, toward the poles. Okay, toward separate poles, where the centrioles are. Then you have chromosomes that are now single chromatid chromosomes. They are at separate poles of the dividing cell. Nuclear envelope starts to reform. This is during telophase, and then cytokinesis happens where an actin bundle of fibers lines up in the middle of these, what's going to become two cells, creates a ring that's going to pinch off those two cells and make two separate cells, and that's called cytokinesis. Okay, those are all animal cells. These are plant cells. You can tell by the plant cell wall. And just to give you an idea of what things look like in plant cells. So plant cells don't have centrioles. They do have centrosomes. So you don't see centrioles in this picture. Mostly this is stained for DNA. So you can just barely start to see DNA condensing, and then it's condensed. And then in metaphase, it's really hard to see them lining up in the middle, but they are. In beginning of metaphase now, it looks more like they're lined up in the middle here. In metaphase, anaphase, the chromosomes are separating. And then in telophase, you get a new cell wall forming between the two daughter cells after telophase. Okay, here's an H5P question for you. Okay, at the connection to the chromosome at the centromere, microtubules are going to connect at the centromere to the chromosome. So here is a chromosome that is in prophase, metaphase, anaphase. Actually, it would be prophase or metaphase, not anaphase. And I know that because it's still two sister chromatids, okay, in one chromosome. And at the centromere, there is specific DNA sequence at that centromere. And that specific DNA sequence will attach to a protein complex called a kinetochore. And that protein complex has an inner layer and an outer layer. The inner layer binds to the centromeric DNA, and the outer layer has this fibrous network that's going to interact with microtubules that attach specifically to the centromere at the kinetochore, and those are called kinetochore microtubules. Looking at more, one more thing there is that in the centromere DNA, that DNA is wrapped around a nucleosome made up of histones, and the H3 unit of the histone gets replaced with a separate protein called CENT-PA, and this is a type of nucleosome that is specific to DNA in the centromere region. So that is one of the ways that this inner kinetochore can attach to the DNA, and then the outer kinetochore attaches to microtubules. So more about how that happens. So this is part of the inner kinetochore. Will have proteins that are connected to that interact with other proteins that form a collar around the microtubule that is the kinetochore microtubule that's going to interact with the centromere. So the plus end of that microtubule interacts with this collar of proteins that is connected to the inner kinetochore. They're not showing you the outer kinetochore on here, which is also fibrous and interacting with microtubules. There's over 50 proteins involved in the kinetochore structure, and the attachment to the chromosomes is secure through those 50 proteins. If you stain specifically for some of these kinetochore proteins, you can see those lighting up on this immunofluorescence stain in these little green dots. Microtubules again would be in red in this case, and then the DNA in blue. So microtubules before have been green, but because they're using green for kinetochore proteins, they stain microtubules red in this particular slide. If you look at a transmission electron micrograph of a section through a chromosome, just to look at the structure of this kinetochore, you can start to see a dark band here, the inner kinetochore, and this light band here, the outer kinetochore. And you can see definitely on a chromosome that is in metaphase or prophase, you have kinetochore microtubules attached to both sides, and they're facing opposite poles of the cell. So this would go in this direction, and that would go that direction, and this would split down the middle during anaphase and go from, which is in this slide, 1 chromosome, but after anaphase, 2 chromosomes. Here's another H5P question for you. Okay, how do chromosomes move around in the cell? There are motor proteins associated with the microtubules. You've seen this before. We talked about dyneins and kinesins walking on the microtubules. There are specific motor proteins for the specific types of microtubules that are in the spindle complex, and the spindle complex is this whole network of microtubules in this color brown in a cell that is going through mitosis. So in metaphase, they're lining up in the middle, and you can see that you have kinetochore microtubules, like right here, that are attached at the kinetochore in green. But you also have some microtubules here that are not attached to chromosomes. And what are they doing? Okay, and then these are the centrioles within the centrosome. Okay, you have motor proteins that are specifically moving the chromosomes on the kinetochore microtubules here, and they're moving them toward the poles, toward the centrioles in this animal cell. But then you also have other motor proteins that are causing these filaments, these spindle filaments, that are attached to the poles also, but not attached to chromosomes, and they're pushing them apart, okay? Pushing these fibers in this direction, pushing these fibers in this direction, and this causes the whole cell to stretch during anaphase. And so you have both movement of the chromosomes in opposite directions, and then you have movements of the whole cell in opposite directions. So the whole cell stretches out. This can be referred to as anaphase A and anaphase B. Anaphase A being the chromosomes moving, anaphase B being the cell stretching out, or pole-to-pole separation. They happen at the same time, so we're not going to worry too much about A versus B. Okay, if we look at just these polar microtubules that are not attached to a chromosome, but instead are attached to the poles, there are motor proteins, motor proteins here, okay, and they work to add microtubule subunits to the plus end of the microtubules, and at the same time, other motor proteins at the centrosome are removing microtubule subunits, tubulin subunits. So as a result of that, that tends to push this microtubule in that direction and this microtubule in that direction. because you're adding on right there and you're subtracting right here. And that tends to, like I said, push apart those microtubules. Since those microtubules are anchored at the centrosome on both poles, that will push the cell apart and generally cause it to widen out in this direction. If we look at the other sorts of proteins that are involved in mitosis and their interaction with microtubules, you have motor proteins involved in 3 different places in the spindle apparatus. So you have at the kinetochore, where it attaches to the chromosome, there are motor proteins that are taking off microtubule subunits on the plus end that is attached to the attached to the chromosome, and then also taking off at the centrosome. So that causes the kinetochore microtubule to get shorter and shorter and shorter in the direction toward the centrosome. So if it gets shorter and shorter and shorter in the direction toward the centrosome, it's going to pull that chromosome with it. And that's what we observe in the cell. The chromosome pulls. And they get pulled that way because there's a motor removing microtubules on one end or microtubule subunits on one end and on the other end, making the kinetochore microtubules shorter. In the what we just talked about in the polar microtubules, these are the ones that stretch from one pole to the other, but they're not connected to chromosomes. There are motor proteins that are pushing against each other. So pushing in this direction in this example on the top microtubule, and in that direction on the bottom microtubule, that pushes the 2 microtubules apart. And since they're connected to the poles, it pushes the poles apart and flattens out the cell in a horizontal direction. And then there's a third set of microtubule proteins that are in the region of the centrosome. They attach to microtubules that radiate from the centrosome, but then they're attached to the cell membrane, and they are removing microtubule subunits, tubulin subunits, from these astral microtubules, causing them to get shorter, and this adds to the stretching effect that happens with the polar microtubules. Okay, so this is the anaphase A and B, the A part being moving the microtubules toward the centrosome. That happens by just making the microtubules shorter. And then the B part, which is stretching out the cell in this, in this direction. And that happens both by these overlapping spindle microtubules being pushed away from each other, and then these astral microtubules here getting shorter, and that pulls on the poles to pull them apart from each other. Okay, these centrioles, which are inside the area of the cell called a centrosome. I know it's confusing because those 2 words sound the same. These centrioles are a special type of microtubule structure. They are triplet microtubules. 1, 2, 3, 4, 5, 6, 7, 8, 9 sets of triplet microtubules, and they're arranged at right angles to each other. We've seen these before when we talked about just parts of the cell. This is what it looks like in cross-section, in a thin section. These are specific to animal cells. Plants don't have them. Plants still have a centrosome, which is the area from which microtubules arise, a microtubule organizing center, but they don't have the centrioles. If you destroy the centrosome, the cell will still make spindle. It'll still form microtubules to make a spindle, and it'll still divide. It's a little screwed up, but it still works. So we don't totally know the role of centrioles and centrosomes even in 2020. This is an electron micrograph just showing how there's overlap in these spindle microtubules in the middle, that they're attached to the poles, and then these motor proteins would be in this overlap region pushing it in opposite directions. Okay, the one on top gets pushed that way and the one on the bottom gets pushed that way, and that stretches out the poles from each other. Okay, so now you've got your chromosomes to either side, and now you want to make two cells from one cell. This is a scanning electron microscope picture. These are diagrams to go along with that. There's a contractile ring that polymerizes in the middle out of actin, and that contracts and pinches off the two cells from each other, forming two daughter cells, each with its own complement of DNA. And mitosis is complete at that point. In about 50 years ago, there was a search for natural products that might be effective drugs, and one was found in this plant, which is the yew tree. It's a tree. An extract of this plant is called Taxol, and Taxol is a powerful anti-cancer drug. The way it works is it binds to microtubules and stabilizes them so they can't depolymerize. And as you saw in the previous slides, you need to take away those tubulin subunits in order for chromosomes to migrate to their opposite poles. And in cancer, one simple way to define cancer is cells that are under abnormal cell cycle control or uncontrolled cell growth. They're not making quote-unquote decisions about when to divide appropriately, and we'll talk about that in 2 podcasts. So they just keep dividing and dividing and dividing. Taxol will target cells that are rapidly dividing and prevent them from dividing, and some of those cells are cancer cells. You do have some cells in the human body that rapidly divide normally as part of health. These are cells that line your gut from your mouth to your anus, because they line an epithelium that gets abraded by brushing your teeth or the food that goes through your gut, and those cells die normally, and you need to replace those cells daily. You also have cells in your hair follicles that go through mitosis routinely, allowing your hair to grow. One of the side effects of these drugs is that cancer patients will have nausea and their hair will fall out, and it's because these drugs target cells that are rapidly dividing. So you knock out the bad and the good at the same time. Modern chemotherapies are more targeted. Monoclonal antibodies that go after a particular protein, bad version of a protein in cancer cells, are particularly effective and becoming more popular. These sorts of treatments are now used in cancers that are discovered late, in that where there has to you have to knock back cancer that is spread to many parts of the body, versus if it's discovered early and you can target a particular cell type, usually you can avoid a lot of those bad effects of chemotherapy. Okay. Here's another cell image where spindle microtubules is labeled red in this image. DNA again is labeled blue, and centromeres, these light colors here. In the case of cells treated with paclitaxel, this is another name for Taxol, then you see that they don't go through, in this case, anaphase. Okay. So in this diagram of a cell that is starting cytokinesis, you see you still have a spindle network. In this case, the microtubule network is labeled in white. But then you start to see a myosin light chain starting up here. So there's an actin ring that polymerizes down the middle. Myosin is used as a motor protein on that actin to actually split the cell in two. There's a protein called Rho, which is organizing the actin subunits into this ring. And then Rho interacts with myosin also to, myosin forms a motor protein that's actually going to cause the ring to contract. So here's staining for Rho on a cell that is getting ready to form a cleavage furrow. And then the cleavage furrow is where the cell is going to go from one cell to two, where that actin is polymerizing, and where myosin will interact with the actin to pinch off those two cells from each other. In plant cells have a cell wall. They're not going to pinch off from each other, but they will start forming a cell wall in between the two cells until it connects to the cell wall on the top and the bottom, and then you have two separate daughter cells. Cleavage isn't always right down the middle. In your cleavage, as you're going through mitosis, as you develop, you have unequal cell division. Here's an example in a frog embryo. You can see in this early stage where you're going from one cell to two, it looks like it's going to result in two cells that are about the same size. But then in later cell stages, you have two giant cells down here, and then much smaller cells up here. So during mitosis, there was unequal cell division, so that not all of the daughters got exactly the same amount of cytoplasm. Some got more, some got less. This is just in a sea urchin embryo how you can see that the spindle apparatus is not set up as you would expect in a cell that was going to just divide in half, but instead looks like there's going to be a division right here, so some unequal cell division there. So why would this be? Why have asymmetric cell division? I want you to think about this, and you may get it as an extended response. So why during development would cells want asymmetric cell division? What advantage does that give? Okay, in prokaryotes, this would be a prokaryote. You have a circular chromosome which is supercoiled here, and you'll have binary fission where the cell is going to divide in two. Instead of actin, you would have a protein called FTZ, which is more similar to tubulin than actin, forming this contractile ring, which is going to split the two cells in prokaryotes. Okay, here's another H5P question for you.